So, Robert, I'm very glad to have you here and listen to your talk.
Cool. Yeah, well, thanks very much for having me. I'm really happy to be here. And I'm looking forward to chatting with several of you this afternoon as well to find out a bit more about what everyone's doing. So today, well, Antonio mentioned that I did elastic capillarity, but what I want to talk about today is actually more about phase separation and trying to control phase separation to make materials with interesting properties. So that could be color or toughness or things like this. I should say, by the way, if you have any questions, please don't be afraid to stick your hand up and ask me instead of waiting till the end. So just to sort of introduce me, so I'm in the group of soft and living materials. I'm a group leader. Eric Dufresne is sort of the professor who's the head of the lab. And what I'm going to talk about today is very much from different people in our group. So the main people, Jin Young, Sai, and Katie, who've been working on all of this stuff. But the main motivation, I guess, for a lot of what our work is color. And so color is something that you probably know is quite hard to keep a good color that really keeps its hue. So if you think of most old colorful things, they might start out with some nice, beautiful reds and bright blues, but over time, typically these will fade and the color will just disappear. And that's because most of the time when we have something that's colorful, it comes from a dye molecule. And that dye molecule basically absorbs a certain component of light. So for example, these green chairs here will have a dye in it that absorbs the blue and the red bit of the spectrum. And what you see is the light will come in, the blue and the red gets absorbed, and then the green bounces off, and that's what you see. But the problem with this is that every time you absorb a little bit of energy from the light, there's a chance that your molecule is going to break. And when it breaks, then you lose that color. So this type of light, typically, this type of color fades over time relatively quickly. And this is also seen when you look at animals. If you have an animal in real life, often it's nice and bright, for example, this fox. But then if you go to a museum and you look at the same fox, an old fox, the color is typically faded to become something much worse. So there are, however, a couple of animals that if you look at a museum, don't fade over time. So butterflies is one famous example of this. And these blue birds here, also, if you look at old versions of these birds, the blue will stay the same color. And this is one of the reasons actually butterflies are very popular to collect, is because they all keep their color very nicely. And the reason for this is that there's no dye involved in making these blue color. Instead, actually, there's a trick that they use. And when you zoom in on the bird wings, you see that they have this structure. So each of these now is showing them a structure of just the blue parts of the wings of these different birds. And these are all made of keratin. But the key here is you can see, well, there's these little air bubbles surrounded by keratin here. But this structure has a very well-defined wavelength. And so each of these bubbles is basically around 100 microns. I feel like this should be nanometers, not microns. Sorry, this is definitely nanometers. But the point here is that all of these structures is almost in the middle of the wavelength of light rate of visible light. So light will come in and bounce off, and basically the way it interacts with the structure actually gives us the color. So there's no dye involved at all. It's just from scattering with a structure that has a very well-defined wavelength. And you can have different types of structure. So now in this one, you have air bubbles, basically, in keratin. In this one, this is, if you know what spinodal decomposition is, or sort of a bicontinuous structure, this is sort of a spinodal-like structure where you have kind of a network that's connected together instead of bubbles. And then up here, you have something that's a little bit more kind of random perhaps. So a couple of things to say is that this is actually interesting because these birds, all of these three structures are made of the same material. And it's a relatively boring material. So the birds actually have this amazing ability to take this material and craft it into a bunch of different shapes. So for us to do the same thing with polymers is actually quite tricky. I'll come back to this in a little bit of time. The second thing to say is actually the birds have amazing control over this, because if you get the size of this structure wrong by about 10 nanometers, you're going to go from something which is nice bright blue to maybe a green instead. And so the birds over their whole wing can keep this structure incredibly uniform. And again, this is something that's very challenging for us. Now, one thing I also want to say is these little insets here are scattering patterns. So if you shine an X-ray through these guys, you will see a scattering pattern. Don't worry too much about it, but the only thing I want to say is that these guys, you can see that they have a ring on it. And a ring in a scattering pattern tells us that we've got something that is pretty monodisperse. It means that it's got a well-defined wavelength that's in that thing. So you can see down here, we've got a really well-controlled thing. We've got a strong ring. Up here, we've still got a relatively strong wavelength, and so we've still got a ring. When we get to the more random structure, you start to lose that ring in the top right one. So the challenge for us, basically, is can we make similar materials to these birds and butterflies? In particular, can we do it over large length scales? And this is the challenge. The length scale of these guys, because it's about 100 nanometers, is really tricky for us. So if it was bigger than 100 nanometers, we could use light, for example, to kind of control UV polymerization. We could make a structure relatively easy. But because this is about the wavelength of light, we can't use any of the kind of the macroscopic 3D printing type techniques to make a structure like this. If it was much less than 100 nanometers, you could use molecular self-assembly mechanisms to start making structure. But this sweet spot around the wavelength of light is actually incredibly hard to make a structure, and especially to make it over a big area. So it's quite a challenge. So to sort of start thinking about this, well, we can think about what are the birds actually doing? And the theory at the moment is that they're actually using a phase separation type of approach. And phase separation at its simplest is if you take two materials, you just heat them up so that they mix together. And when you cool them down, it will do phase separation. So it will make droplets, something like this. And the idea being, can we take a phase separation process and make a material like this? So that's all well and good in principle, but the tricky bit comes actually when you start trying to do phase separation. And so this is just a demo where it's two liquids that they mix together. Actually, when they're cold, when you heat them up, they start to phase separate. And this is just, I think it's probably 500 microns across on a microscope here. And we're just going to heat it up and see what happens. And so you can start to see the phase separation occurring. And as this comes round, maybe if I pause it. Oh, no. Sorry. In the very initial stages of phase separation, if you look here, you can actually see that we've got really quite a good wavelength coming out of it. This is of a nice uniform material. But the problem is that as this starts to evolve, these little droplets of liquid that are sort of hanging around in the other one, they start to coalesce. And so this nice wavelength that we have at the start starts to get rougher and rougher. And towards the end, you really see, no, sorry, having an error. It's about 500 microns from one side to the other side of the video. So yeah, you can see on the right-hand side now, you've got like a lot of mess that's starting to come out. And so the reason we're losing this is because these droplets of liquid, they're just coalescing with each other, and it just coarsens the pattern. There's another thing called Ostwald ripening that I'll come back to too, but in this case, it's not really super relevant. So what we wanted to do is, well, we want to keep this nice uniform length that comes out at the start, but not have all of the coalescence that's causing us this mess at the end of the phase separation. And so what we thought is, well, why don't we just add in a polymer network? So if we take a gel, this is now a polymer network swollen with one liquid, we can add another liquid in to make it a combination of two liquids with this polymer network. And then when we drive phase separation, the idea we're going to get droplets, but because they're embedded inside of this polymer network, they can't move around and coalesce with each other. And so ideally, this was going to give us something nice and uniform. And the way we do that is you just take something like a soft silicone gel, PDMS, for example. You dunk it inside of another liquid that it's partially soluble with, heat it up in the oven overnight so that it diffuses into the gel. And then literally all you do is take it out and put it on a microscope and see what happens. And it looks something like this. So these are now fluorinated oil droplets that are just growing by phase separation inside of the gel. And at the end of the movie, you can see that actually with very little effort, so literally all I've done is taken it out of the oven and put it on a microscope, we've got something which is really quite well, not completely monodisperse, but pretty close to being all of the same size droplets. And this is just 100 microns, but we can actually get this on the scale of centimeters very easily. Just to say, this is, by the way, is a confocal image. And what this particular type of microscopy, Carr's microscopy, allows you to do is see where the polymer network is. And so what we're seeing is that inside of the droplets, actually all of the polymer network is just excluded. It completely pushes it out. So you've just got a pure liquid droplet surrounded by your polymer. And then this is just sort of scanning up and down through the material, again, to show you that you have something that's pretty monodisperse. So I mentioned earlier about these rings. If you shine a laser through this, you effectively do a scattering experiment. And we can start to see that we've got a little bit of a bright ring going on here, which is telling us that we're actually doing a pretty good job of making a monodisperse material. But more sort of visually, if you shine a white light through this, you start to see color. And this is coming because of this idea is if you have monodisperse things, light will interact with it, and different wavelengths interact differently. So you start to see the blue light being bent really well, and then the red light in the middle, it just comes straight through. So you start to see these rings of color. So this is a really good sign that we're starting to get sort of monodisperse stuff happening. You can also, because this is in a stretchy material, if you stretch it, you see the color change a little bit. So this is not a super strong thing, but again, sort of ultimately we were interested in seeing, could we have a material that would change color as it stretched? And this is because the little inclusions inside of this are changing shape slightly as you stretch the thing. So I showed you we could get pretty monodisperse droplets. We can also change the size of these guys with a few different parameters. So for example, cooling faster or slower, you can go from something which is a bit bigger in terms of the droplet size. And when you cool these guys much faster, you get tiny little droplets. And so with this cooling rate, we can change this by, say, an order of magnitude, maybe 1.5 orders of magnitude. So in this case, these guys are about 10 microns, and we can go down at the lowest to, sort of 600 or 700 nanometers from memory. We can also tune these guys a little bit with two other parameters. One is the stiffness of the gel, because we have this polymer network. We can tune essentially how dense and how cross-linked that is. And if you increase the stiffness of your material, you get smaller phase-separated droplets. And for softer materials, you get much bigger ones. And at the same time, we can increase how much liquid there is. So if we add more liquid, you get bigger guys. And so with these three different parameters, we've got pretty reasonable tuning of these inclusion sizes over maybe 2 orders of magnitude, I would say. So the stiffness is interesting because that also allows us to play a few other tricks. For example, if you have a gradient in stiffness, this is now work from Katie, is she put a soft gel on this side, a stiff one on the other side, and it allows you to start tuning where your droplets actually grow. Because what happens is over time, the ones in the stiff side dissolve and then they move towards the softer side of the gel. And you can see if you look at the ones right at the interface, these actually grow because the liquid from in here is actually being squirted into these ones at the interface. So by controlling sort of stiffness gradients or local cross-linking densities, we can actually control where this phase separation is going to happen. And then, like I mentioned, if you stretch the stuff, you also control the shape of them. So if you have a round droplet, when it's not stretched, you put a stretch on this guy, you start to see these things forming little ellipses. So I'm not going to just mention this in passing. What we were really interested in with this is a nice way to kind of investigate how small cracks grow in materials. So if I have a piece of an elastomer, you can think of these as being a tiny little crack that's nucleating inside of the elastomer. And because we can grow these super slowly, we can actually look for bond breaking around the edge of these things and see how the network breaks. Okay, so why does this technique work? And there's really kind of two key features that are important here. And the first is if I show this video again, and you look at when the nucleation happens, you see right at the start, there's a burst of nucleation when all of these droplets grow for the first time. And then after that, you don't get anything new growing. So there's a sudden burst, and then nothing happens after it. You can see now there's all of the burst of nucleation. So this is the first key point that's important for understanding why this works. The second thing is that the droplets don't like to be near each other. So if you plot g of r, so this now is a function that basically says, if I have two droplets, what's the likelihood of finding them a certain distance away from each other? So if the droplets are a large distance away from each other, basically they've got a g of r of 1, which tells me that there's many droplets at that distance. But this function drops off as you get closer and closer, which is telling me that the average space, there's typically a gap around every droplet where no other ones are there. So the droplets tend to be spaced apart as well, is what this is saying. So why is that happening? What we think is going on is when you nucleate a droplet, it does two things. Firstly, it locally depletes the polymer around it of droplet phase, and that makes it harder for another droplet to appear nearby. Also, because this thing is pushing open its hole, it's compressing the network, and that also makes it harder for another droplet nearby to nucleate. So these two effects basically stop another droplet nucleating near this guy. So you get this burst of nucleation, all of these guys are spaced apart, and what you can then think of is, well, if I make a Voronoi cell, basically draw lines around each thing, each droplet, and say everything inside of this cell is closer to this droplet than to any other droplet. And just say, well, all of that liquid goes into this guy to grow it. If you do that calculation for our experimental system, it seems that this is basically what's happening. So you randomly nucleate your droplets, they're all spaced apart, and then they just suck in liquid from their surroundings. And by the time they've grown, they turn out that they're pretty much all the same size. So that's all that we think is going on, but it seems to work in a pretty nice way. So I was going to skip that one. This was one type of phase separation controlled by temperature, but there's many different ways you can phase separate stuff. So another type of phase separation that you might have come across is Ouzo. So Ouzo is basically a mixture of ethanol and oil, so anise oil. And when you drink Ouzo, you put water in with it, and water turns this Ouzo cloudy. So I think there's also pastis in France that you guys are looking at. It's the same principle, right? And the idea is that the oil is soluble in ethanol, but when you add water into it, because oil doesn't like to mix with the water, it phase separates into oil droplets that are suspended in kind of a water-ethanol mixture. And because this whole principle that I was talking about with the phase separation doesn't really rely on kind of any particular mechanism, this type of phase separation should also work. And so we tried this. You basically take a hydrogel, you stick it in your Ouzo solution, which, by the way, is very smelly. You don't want to work with this for too long. And then once this is saturated with Ouzo, you drop the whole thing in water. Now the water diffuses in. And the idea is you see if you're going to have something similar. And indeed, you get the same thing with little droplets of oil forming inside of your hydrogel. Yeah, I don't want to try to. This is edible, actually, because it's both. We were talking about you could make jelly babies that are stretchable and they change their color, which would be pretty cool. But yeah. Okay, so I've shown you we get this type of phase separation, but what the birds have is still very different, right? So we have this really dilute network, dilute number of droplets. They're much more dense, and are actually a little bit smaller than what we see here. And so if we look at the color, we're seeing something which is much weaker than what the birds have. So what can we do to get closer to this type of structure? And Alba has recently been working on doing this with a different way of phase separating again. And so what she does is she takes polystyrene, so just the plastic, and then you swell it with MMA. So this is PMMA, is also a type of plastic. This is just the monomer for polystyrene. Now, this swells polystyrene. It turns it into this sort of, it's just from a plastic into this sort of soft, squishy mess. But when you start to polymerize, you take these monomers and you polymerize them. Now, because these are getting longer, they don't want to be, the entropy isn't favorable to keep them dissolved in my polystyrene network, and they start to phase separate. And so what you get is these little blobs of PMMA that are inside of your polystyrene network. And because this is essentially just the same mechanism, again, we've got a polymer network, and we've got phase separation inside the polymer network, we, again, get this nice type of phase separation with this sort of blue color. So this is now just showing the initial amount of monomer that I put in the sample. So this is initially 14% of monomer in here, 21 and 38. And you can see that we get this nice strong blue color coming because of this phase separation. The point of these guys is one, just to show that we can do it over large areas. So we could go larger than this, but we just think for practical reasons, this is big. And then we were in collaboration with this company who was interested in making fibers. And this was just kind of a proof of principle to show that you could potentially make fibers. So I think they wanted to have fibers for, if you could have some cloth, for example, that doesn't change color over time. This would be nice, because then you could make things like curtains that would stay the same color. So, yeah, no. I'll tell you about that in maybe 3-4 slides, but it's a very good observation. The trick here is actually that these are on a black background. If these were on a white background, they would look a different color. And I'll explain why that's important in just a sec, but it's a great question. So the structure, this is the bird feather structure. And now this is just a scattering plot, which shows this kind of a nice peak. Our structure is starting to actually look much more like this bird feather structure. It's got sort of these inclusions. What's a bit different is they're a little bit more blurry. So the edges of the inclusions aren't quite so sharp. And that's why we think we're not getting such a strong color as the blue bird color. We're getting this slightly sort of paler blue. But if we compare what we have in terms of scattering to some other birds, we're really getting towards what nature is doing. And so this is kind of a good sign, I would say. I'm just going to skip over that one and come to your question. So a trick that with structural color most people don't really talk about is actually you need one extra thing to make nice color. So this is now the sample, one of the samples that's phase separated on a black background at the top and on a white background at the bottom. And you can see that on a white background, the thing just looks white. So we don't have any of that color. You need to have a black background behind your material if you're going to have good structural color. And so the reason for this is, if I imagine what's happening to my lights, I've got my blue, green, and red lights coming in. Red light will, in this case, not be scattered very strongly by the structure. And so it's just going to go through the sample. Green light will scatter a little bit, and then blue light is going to scatter very strongly. And so the function of the black background is basically to absorb all of this light that's coming down and going through the sample. And then all you see is the blue light that's coming back up. If you have a white background, now if this is white, this light can actually scatter back up. And so all the light ends up coming out of the sample, and it still looks white. So in order to get structural color, you actually need an absorber to remove the color that you don't want. So this is a bit awkward because also what happens is if I have a very thick piece of material that has my structure in it, all of the light will just still scatter. So the red light will scatter weakly, but eventually it will scatter and come back up. So a thick piece of really nicely structured material will also look white. It won't look blue. But there's a trick that you can do. If you add some absorbing black particles, some carbon black, for example, what will happen is now the blue light will come in, and before the carbon black has time to absorb it, it can get scattered out of the surface, and you get that blue light back. But the other light, it'll go down. It will slowly scatter, but because of these black particles, that it will be absorbed before it can come back out the top. So if you take something like now a piece of polystyrene and you add in some of this black particles and now make the structure on top of it, we do actually start to get blue materials. And that type of material doesn't look white if I stick it on a white background because it's got the absorber inside of it. So just to kind of make the point, I think I really like this picture. So this is a bird. It's got a nice blue structurally colored bottom, and then the top is black. And this color comes from melanin. So the same pigment that I think we also have melanin when you tan. The bird on the right is an albino bird. The only thing that's different is it can't produce melanin. So it can't make black color. And you see that all of the color is gone. It's not just the head where it was black has gone, but also the body. This bird still has the same structure that makes the blue, but because it's missing this absorbing part, it can't make the color like it would otherwise. I hope that answers your question. Probably more detail than you needed, but okay. So we're starting to get closer. Another thing that I mentioned at the start was, well, birds can also make this bicontinuous network type of structure. And so can we get anything that looks like that? So what we think is that this is actually a spinodal decomposition structure. And when we're doing our phase separation with just a little bit of fluorinated oil in it, we get this, what you might call nucleation and growth. It's just too dilute. So what Carla has done, and this is super recent work, is upped the concentration. She basically found a different monomer that swells this gel like crazy. So this goes from, it basically swells it so there's 80% liquid. And then she can drive phase separation in the gel from the highly swollen gel. And actually what she sees when she does this is you get something which looks exactly like this sort of spinodal decomposition structure. And the scattering pattern is this beautiful ring. So with this, we think we can actually start to reproduce some of these structures like the birds have here. What is also cool is because the liquid that she's using to drive phase separation here is a monomer, she can then polymerize it. And so now you have a structure with this bicontinuous network that's locked in. And so you can not only look for things like the color, you can also do mechanical tests on it and see, does it have interesting mechanical properties? And the answer is yes, actually, it has surprisingly good, for me, amazingly good properties. So now this is basically showing this is an elastomer that's swollen and then polymerized. If I just look at the elastomer, the original elastomer that I made, it's pretty stretchy, but pretty soft, and then it breaks very suddenly. If I look at the second phase, so this is a monomer that I've polymerized and turned into a plastic, this is really brittle. It's pretty stiff, so you get a high slope at the start, and then it breaks at a tiny strain. But if I now swell my elastomer with the monomer and then polymerize it and look at the mechanical properties of the composite, I get something which is more stretchy or more stretchable than the original material. And the toughness of it, so if I think about the area under the curve, is huge in comparison to both of them. So somehow this combination of a plastic and the elastomer mixed together with this bicontinuous structure is really giving me this sort of synergetic properties.
Just a detail, but I'm confused by the scale for the scattering pattern.
Oh,
and reciprocal microns, I guess.
that looks about, yes, that's yes. 10 nanometers. It's light scattering, yeah. Okay. Yeah, so at the moment, we're not entirely sure what's causing this good properties. We know that with a lot of materials, you can make double networks. So if you guys have come across double network gels, for example, by combining two networks together, you can get these toughness effects. What's interesting here is that we didn't have to try very hard to get this to work. So normally when you have two materials and you put them together into a composite, you have to tune the ratios really precisely to get it to work. In this case, it always seems to work well. So we think that there's some interesting physics there, but we just haven't got to the bottom of it yet. Okay, so the last thing I want to mention in terms of making these sort of permanent materials is making crystals inside of a network. And so from what we were really motivated by here is I'm in a material science department, and the material that everyone wants to be able to replicate from nature is nacre. So if you've come across this mother of pearl, inside of the clamshells, they have this very beautiful material that's incredibly tough. And that material is basically made of little crystal platelets of calcium carbonate, little crystals, and they're surrounded by a soft material. So what we wanted to know is, could we make something that looks similar to nacre, so little platelets of calcium carbonate inside of a soft gel? And so what we started off by doing was, you can take a hydrogel, in our case, PEGDA, it's just a generic chemical hydrogel. You swell it with a calcium chloride solution, and then next to it, you put sodium carbonate. And the idea is that the carbonate ions, they diffuse in, react, and then you form calcium carbonate crystals. And if you do that with different concentrations of your solution, you can see that you get these particles, these little crystals of calcium carbonate forming. And again, they're pretty nicely the same size as each other. So the same mechanism is working here, because we're having phase separation inside of a polymer network. And as you increase the concentration of solution, obviously you get more and more of these things. The issue here, though, is that ultimately this is diffusion controlled. So we've got this bath of ions here that have to diffuse in. And basically what this results in is a lot of crystals that are forming close to the interface, and then not much that's forming away from the interface. So we're not getting a homogeneous material, is what we'd ideally like to do. And so Damien Palin from Cornell, who we've been working with, solved this problem by making a new geometry where now you have a hydrogel, and he used agarose. You put one of your solutions on one side of it, one of your solutions on the other side, and if you have a thin layer of hydrogel, these guys will cross diffuse, and you get these particles forming in the middle of the thing. So this is just to show you what the hydrogel looks at the nanoscale. So you've got a pore size which is sort of order 100 nanometers. And then this is what the crystal looks like when you dissolve the hydrogel network and then bring them out. So calcium carbonate has a sort of a cubic structure, which is why these guys look like little cubes. It's also got, you can see they're a bit pointy on the ends here. And what's happening is, because diffusion, this is diffusion controlled, diffusion can happen faster to the points of these cubes. And so you grow a little bit faster at the edge. And it gives you what people call a hopper-like structure. So what Damien found, though, was that he could change his hydrogel from something that's nice and isotropic like this to something that's actually kind of directional. And so what he did was, he took his hydrogel, he dried it out, so it's now completely dried, and then you re-swell it. And when you re-swell it, it doesn't come back to its original thickness. It comes up to maybe 50% of its thickness. And all the fibers inside of the hydrogel sort of align perpendicular to the drying direction. So now I've got something where the fibers are all aligned like this, and my drying direction is in this pink line here. And what's interesting is, when you make particles, or when you make crystals inside of this thing, you switch from, in the original case, we had these little cubes, essentially, to, in the dried and rehydrated sample, you get something which are now platelet-like. So they completely change their aspect ratio. And so this gel actually allows us to kind of shape these crystals a little bit to form something like platelets, which is ultimately what we were looking for this nacre problem. You can also image these in situ, and what you see is, now this is the drying direction up here. So my fibers and my hydrogel are aligned in this direction, and the platelets, they look like they're trying to grow parallel to these fibers with this big aspect ratio. The next step for Damien was to try and dry this out. So the idea is, as you dry your hydrogel out again, it would compact all the platelets together. But this is still a work in progress. So this is, I would say, as far as we've got at the moment. So still have this dream of making it work, but we're not quite there yet. Okay, so what's going on? Why is it forming these kind of platelet-like structures? And what we think is happening is, when you start pushing open a hole inside of a network, initially you can push open that hole in a pretty uniform way. And there's this thing called a cavitation instability. Is when you push open the hole, it doesn't get harder and harder to push open a hole in a polymer network. It reaches a certain pressure, and then it can just grow without changing the pressure. So actually, it's not as hard as you might think to push open a hole inside of a polymer gel. But at some point, you're going to start fracturing. And so you're going to create some localized damage on your hole. In this case, this is what I've sort of drawn here with the red. And as soon as you made that little damage at the end, the thing is going to grow like a crack. And that crack is going to have a big high aspect ratio, like our platelets, with the length is going to be much bigger than the width. So, oh yeah, and this is kind of just to show what it looks like. Initially, if you pump open a hole inside of PDMS, it's pretty round. And then in a matter of 1 or 2 seconds, suddenly it will break somewhere and you sort of develop this crack tip. So there's this transition from something that's round to something that's going to start being high aspect ratio. And now you can use results from fracture mechanics. So fracture mechanics actually says, well, the aspect ratio is going to go like the length of my crystal divided by this length scale. So gamma over E is a material length scale. So gamma is the fracture energy of my material, and E is the Young's modulus. And if I combine these two together, I get a length scale. And I can stick it in here. I can measure this from bulk properties, so I can tear my hydrogel and measure the fracture energy. I can measure its stiffness, and that gives me one value, so this is 24 microns. And then from the microscopic guys, I can measure L and W, and that actually gives me another measurement of this parameter, and I get 10 microns. So we get pretty good agreement between these two things, which make us think that this is really the process that's kind of allowing us to shape these little platelets. So I think that's everything I wanted to say about phase separation in networks. The other half of our lab, we do a lot of physics in our group, actually does biology. And I just wanted to mention that there's actually some strong ties to what's happening in biology with phase separation too. And this is because inside of living cells, there are a ton of little phase separated droplets. This is just showing a couple of the different types. But if you haven't come across them before, these are interesting because in our cell, your cells are constantly doing chemistry to stay alive. And so that chemistry involves, two materials coming together and reacting. But one of the points of these phase-separated protein droplets is that imagine I've got two chemicals that are floating around inside my cell, but one of them preferentially wants to go inside of these protein droplets. What will happen then is, let's say I put it inside my stress granule, one of those chemicals is kind of contained, and because it's contained, it's not going to be reacting with the rest of the chemical that's surrounding it in the rest of the cell. What the cell can then do is if it dissolves suddenly my stress granule, it will flood the cell with a bunch of chemicals that then can start up that reaction, allowing the cell to do something very quickly. So the cell can basically turn reactions on and off incredibly fast just by controlling the phase separation of these little protein droplets. So this is probably the most famous example of these protein droplets in a cell that's about to divide. And the green dots here are P bodies, so protein droplets essentially inside of the cell. And you can see that the cell has pretty good control over where these are. But the main thing I want you to take away from this video is actually the cell is not a bag of liquid. You can see here all of this structure, and this is the cytoskeleton, which is essentially a polymer network. And these droplets are living inside of this polymer network, which is essentially what we've been looking at with the kind of the physical system. It's just a much more complicated version. And so the biology guys in our lab are basically trying to make the parallels between this system and what we've been learning from the polymer physics. And just to give you a little taste of that, so now this is a work by Tom, where he's staining the cytoskeleton here. And so the red is basically the cytoskeleton, this big black hole in the middle is the nucleus. And you can see that there's a couple of little holes in the network. And when he stains some stress granules, which is a type of phase-separated droplet, the position of these stress granules is basically where these dark holes are in the network, showing that these droplets are kind of essentially growing a bit like the picture that we've been thinking about with our system. If you're interested, have a look at this one. There's also some interesting physics because the cytoskeleton is essentially fibrous networks. And there's also some interesting physics about how fibers interact with droplets and surface tension. Okay, but I think that's everything I wanted to say. I went a little fast, but in conclusion, We want to control phase separation, basically, to stop it from coalescing to make useful structures. And the way we do that is simply by adding a polymer network on top of the phase separating system. And with that, actually, you can get pretty nice color. So these sort of blue color. And what's important is you can make this easily over large length scales. So Alba, who made this thing, her dream, she was telling me, is to be able to have some sort of 3D printer where you print and phase separate at the same time. And if you control your phase separation, you can actually potentially control the color as you're extruding your thing. So you could get blue or green all from one system as you print it out, and it won't fade. So we're still a long way away from that, but it would be pretty cool if we can pull something like that off. Basically, the polymer network seems to control all of the growth, and so understanding this link with the physics for us is kind of like an interesting challenge. And then, like I said, there are ties to biology systems, and a lot of people have been thinking about these phase-separated droplets. But a big part of this is actually how these phase-separated droplets live inside of the cytoskeleton. So, on that note, thank you very much for your attention. And if you have any questions, I'm happy to take them.